let's welcome.
Okay, thank you very much for your introduction. Thank you very much. So I'm a retrovirologist and immunologist, and I have been interested in retroviruses since I was a medical student. And so today I'm talking about the story of the arms race between retroviruses and the us, mammals. Okay, so I don't know how many of you know about retroviruses, but the retroviruses are the name retrovirus, meaning that the retro is going backward. And the name retrovirus, meaning that the reverse transcriptase containing virus. The retro is the abbreviation of reverse transcriptase containing. So what is reverse transcriptase? Watson and Crick stated that the genetic information goes only this direction, from DNA to RNA by transcription and from RNA to proteins, polypeptide by translation. But it was found that there are viruses that contain RNA as a genetic material and convert RNA into DNA by the process called reverse transcription. There's a special enzyme named reverse transcriptase that uses RNA as a template and produce double-stranded DNA. So the retrovirus is very, very special because it goes against the central dogma on the flow of genetic information. So this is what retrovirus look like. It's a very small virus containing 2 copies of single-stranded RNA as a viral genome. And the viral genome is covered by viral proteins. And it has an envelope produced from the cellular membrane of previously infected cells. So it comes out from the infected cells being covered by a part of cellular membrane. And the cellular membrane contains a viral protein named surface protein that works to bind onto the target cells to get into the cells. Okay? The viral genome is about 8 to 10 kilobase. And it contains mainly 3 structural genes. One is GAG that produces the internal proteins that covers the RNA genome. And the other is called POL gene, that is polymerase gene, and that encodes the reverse transcriptase. That is the secret of the retrovirus to replicate inside the cells using the RNA, conversion of the RNA into the DNA. And envelope gene encodes for the proteins on the surface of the virus. And the mechanism of reverse transcriptation is as follows. The viral genome is a single-stranded RNA, but inside the viral particle, the retroviruses contain not only the single-stranded viral genome, but also the single-stranded RNA tRNA, that is fetched from the infected cells. So the retroviruses use the transfer RNA of the infected host cell as a primer to start producing DNA. So reverse transcriptase use the tRNA as a primer and started to produce DNA, single-stranded DNA of minor sense using this tRNA as a primer and goes this way from 5' to 3'. So one secret of the retrovirus is the presence of this R sequence. That is the repeat of the exactly the same sequence on the both sides of the RNA genome. When the DNA is reverse transcribed into the R sequence, it jumps back to the 3-terminal end of the same sequence, R sequence, and this is used as the primer to continue producing the negative strand DNA to this area that is or also hybridizing to the tRNA. This is a primer-binding sequence. And then after that, this RNA chewed up and leaving only the small portion of the viral RNA at the second primer to produce the positive strand. This portion is called polypurine tract because it contains a lot of purines in it. And when the positive strand is extended to the primer RNA, it produces the primer binding sequence here, and this positive strand jumps again back to the 5' primer binding sequence, and from there, positive DNA strand is extended all the way down to the end of the 3' end of the viral genome. So this process produces the exact repeat of the 5' sequence at the end, 3' end of the positive strand DNA. And this repeat of the same sequence, the same set of sequences composed of R sequence and U5, 5' unique sequence and 3' unique sequence produces the exact same repeat of the 3' U3R and 5' sequence. And this sequence works as a very, very strong promoter to express the DNA sequence and transcribe it into messenger RNA. And this double-stranded RNA, when the virus is replicating in the infected cell, is integrated into the host cell genome. So this is the life cycle of retroviruses. The incoming virus particle first attaches on the surface, the cell membrane of the target cells using the surface protein to bind the cell surface molecules as a receptor. And when the viral membrane is fused with target cell membrane, the mangan or magnesium ion is going inside the viral particle, and it switches on the reverse transcriptase. And reverse transcriptase, as I told you, produces the double-stranded DNA from the single-stranded viral RNA genome, and this double-strand DNA is then transferred into the nuclei and circularized, and then by the function of part of reverse transcriptase named integrase, integrated into the DNA genome of host cell. So what integrase does is make a nick shortcut on the infected cell viral DNA and insert the viral DNA into the chromosome. So once the viral DNA is inserted into host cell genome, retroviruses can replicate in two different ways. One is infected cell, when infected cells divide, the infected cell replicate the host cell genome itself, including the integrated proviruses, the integrated viral DNA is called provirus. And so the retrovirus can replicate by hiding into the infected cell genome, and it can replicate depending on the dividing potential of the infected cell itself. And the daughter cells produced from infected cells already contain the proviral DNA integrated into the viral genome. But as I told you, the retroviral the DNA contains long terminal repeat that contains very, very strong function of DNA transcription promoter. In the infected cell, when this promoter works, it produces messenger RNA. Large number of copies of messenger RNA is produced from one integrated viral DNA. And interesting thing is, for the retroviruses, messenger RNA itself works as viral genome. So this messenger RNA, of course, directs the production of viral proteins, and the messenger RNA itself is covered with produced viral proteins, and the retroviruses, the progeny viruses, comes out from a cellular membrane. The way of the production of progeny viruses depends on a so-called budding. Budding means that the infected cells produce virus particles by pushing the virus particle out from the cellular membrane, and no disruption of cellular membrane occurs during the process of budding, meaning that once cells are infected with retroviruses, those cells can continue producing virus particles without being destroyed. Okay? So that is why retroviruses are so difficult to handle, to fight against. Once you are infected, for example, with HIV, you have your own cells that contains viral genome and that continue producing viruses without being destroyed. Okay? Other viruses, especially large DNA viruses, comes out from the cells by producing large number of cellular particles inside the cytoplasm and destroy the cell membrane. So infected cells are destroyed, or most cases, when it releases. They release viral particles, but in the case of retroviruses, the budding of the virus does not affect the viability of infected cells. So, as I told you, retroviruses contain reverse transcriptase and convert RNA genome into DNA and integrate it into infected cells. If this integration occurs in germ cells, like cells that produce sperm or ova, okay, it causes the presence of the viral genome in the sperm or egg, and when the infected sperm or egg is fertilized and produce the embryo, it causes that the presence of the integrated provirus in all the cells of the progeny individuals, right? So it's called endogenous retroviruses. So in the history of mammalian evolution, we have been attacked by incoming retrovirus many, many times. And during the history of millions of years, we had experienced the repeated wave of retroviral attacks several times. That caused the presence of endogenous retroviruses throughout our genome. So the most unexpected discovery made by Human Genome Project was that up to 45% of our genome is actually filled with endogenous retroviruses that integrated into our genome from millions of years ago. And about 8% of our genome is actually consists of LTR-containing retroviruses that can make infectious particles if it is expressed, okay? So the consequence of that is very, very severe because the retroviral integration disrupts the integrity of our genome. If the retrovirus is integrated in a functionally important human gene, it disrupts that kind of functional gene and causes diseases. As a matter of fact, many of known diseases of ours is associated with the germline integration of the endogenous retroviruses that happened millions of years ago. For example, hemophilia or X-linked A-gamma globulinemia or X-linked SCID, those severe diseases, or some breast cancers, all caused by the mutation, disruption, or change of the expression of our genes caused by the integration of retroviruses. And of course, newly infecting retroviruses can cause the somatic cell mutation. For example, if you infect newborn mice with Moroni murine leukemia viruses. So Moroni murine leukemia virus is a simple retrovirus that can replicate by the process as I told you, but this virus can replicate inside the infected cell and produce new virus particle, and new virus particle go into different cells and as a consequence of repeated infection, the integration site continue to increase and finally hit some gene that is associated with cell proliferation or differentiation and cause lymphoma or leukemia. Okay, so the mechanism of induction of leukemia by retroviral infection is not because the viruses contain cancer-inducing genes. Okay, some viruses actually contain the gene that induce the proliferation of cells that work as, for example, growth factor, that work as the molecule that inhibit immune response. But in the case of retroviruses, usually, retroviruses do not contain any cancer-causing gene itself, but the integration of retrovirus in the proximity of, for example, cellular gene that induce proliferation causes the increased expression of that particular gene, and that is associated with the induction of cell proliferation or lack of, for example, differentiation. That's called promoter insertion, or enhancer activity can be observed. For example, the retroviral LTR can not only work as promoter, but also affect the expression of adjacent genes by showing the enhancer activity. So this is how the infection of retrovirus and the repeated integration of retrovirus can cause lymphoma in mice. This process is not necessarily deleterious. It's known that we were actually utilizing retrovirus to acquire new genetic ability to express our gene in different places. For example, amylase. Our ancestors, the primate ancestors, contained only one amylase gene, but this amylase gene was duplicated throughout the evolution, and the amylase gene copy numbers of amylase gene increased during primate evolution. And one of them contained integrated retroviruses, and the enhancer effect of integrated retrovirus changed the pattern of expression. So our ancestors expressed amylase only in the pancreas, but after the integration of endogenous retroviruses, our recent ancestors start to express amylase in a salivary gland. So we have to thank retroviruses that we can enjoy sushi because of expressing amylase in a salivary gland. So this is an example of how retroviruses can change our genetic information and the expression of genes throughout our body. So to protect the integrity of our genome from repeated retroviral attack, of course, we have acquired ways to register against retroviruses. One example is FB4 gene. This is, I'm talking about the mouse studies of retroviral restriction factors. So one example of the gene that has been acquired to register against retroviral infection is ironically, the endogenous retrovirus itself. So endogenous retroviral envelope gene produce a protein that is is expressed on the surface of infected cells. And since this is an envelope gene product, it interacts with the cellular receptor for the incoming retrovirus. So it means that this endogenous retroviral product compete with the attachment of newly incoming retrovirus on the surface of the target cells. So this is one example of how we acquired the resistance factors against retroviral infection. Another factor is mutation of the surface receptor that is associated with the proliferation, growth induction of infected cells. And another example is FB1 gene that is found in the United States by a researcher named Frank Reilly. And the mechanism of action of this gene, FB1, is identified by a British researcher, Jonathan Stoy, who is a good friend of mine. So this FB1 is also endogenous retrovirus. And the product of this FB1 gene interferes with the transport of reverse transcribed DNA into cellular nuclei. And ApoBex3 is also a restriction factor which mammals have acquired in a long period of evolution. And this is an enzyme. And this enzyme is this enzyme interferes with the process of reverse transcription and the transformation, reverse transcription of RNA into DNA is hampered in the presence of this protein ApoBex3. So among these different mechanisms of mammalian cells that interfere with the replication of retroviruses, ApoBex3 is by far the most effective, and there are a lot of evidence supporting that during the evolution of mammals, ApoBex3 has worked as the strongest restriction factor against retroviral infection. So what is ApoBex3? ApoBex3 is cytidine deaminase. And in the first infected cells, the retrovirus, as I told you, reverse transcribed the RNA genome into DNA and integrate it into the cellular chromosome. And when it is expressed, it produces viral messenger RNA, which works also as viral genome. But in the infected cells, ApoBex3 protein is also produced, and it integrates into the budding viral particles. The mechanism for the integration, mechanism for the carrying of the ApoBex3 gene to the viral particle is that this ApoBex3 protein interacts with the viral RNA and going into the budding viral particles along with the viral RNA. And ApoBex3 works not in the initially infected cells, but it works in the next target cells through this mechanism. Okay, so the ApoBex3 goes into the next target cell of infection along with the viral RNA. And when the reverse transcriptase makes the DNA copy from this RNA, ApoBex3 works on the first strand of DNA. The viral genome is a messenger RNA, so it is a positive strand, but the first strand of DNA is a negative strand. So ApoBex3 is a cytidine deaminase. It deaminates cytidines into uridines. And as a result of that, the first negative strand DNA contain C to U substitution. However, as you know, uridines are only found in RNAs, not in DNAs. So if our cell finds that there's an uridine within a DNA. It removes U from U base from the produced DNA, and a basic site is produced, and the A basic site is a target of repair by cutting the DNA strand. So it causes the destruction of first produced negative strand. And at the same time, if this C to U substitution is not removed by the DNA deglycosylase, it works as it is utilized at the template to produce the positive strand. And U is hybridizing with A, so meaning that the positive strand DNA will contain A instead of the original G. So there's a G to A hypermutation occurring in the positive strand viral DNA. So it causes the mutation from G to A at a very high frequency. And when G is substituted to A, it causes the amino acid substitution and the protein is not functional, or it causes the insertion of the stop codons in the positive sequence, positive strand DNA sequence. So this is how ApoB3 works to fight against the retroviral infection. However, we are fighting not with one retrovirus species, but also we can be infected with many different species of retroviruses. And there are a type of retroviruses that can come out from one species of mammals and infect different species of mammals. For example, there are viruses, retroviruses called Xenotropic viruses that is produced from monkeys but infect rats or cats. Or xenotropic viruses can be produced from mice or rats and infect humans. So we are always facing the infection with retroviruses that are coming from different species. So because of that, mammals have acquired the repeated copies of ApoB3. For example, mice have only one copy of ApoB3 gene on the chromosome, but cat has 3 repeated copies of ApoB3 gene, and horse has 7 copies, and humans have 8 copies of ApoB3. And when we have the duplicated number of ApoB3 genes, we have room to have mutations in some of the copies so that we can fight against the attack by newly incoming retroviruses. So, so far so good, but retroviruses are also evolving because retroviruses has to survive by compete with the restriction mechanisms expressed by the target animals. So, and the worst thing is, retroviruses are evolving much faster than us. So our DNA polymerase is strongly error-prone, and our DNA polymerase makes only one mistake in 1 million base pairs. And if a DNA polymerase makes a mistake, we have a repair mechanism and excise the paired nucleotide and make a nick and replace the mistaken nucleotide with the correct one. But in the case of retroviruses, the reverse transcriptase is error-prone, and it makes one mistake per 1,000 bases, meaning that the retroviruses can mutate 1,000 times faster than us. And of course, most of the mistakes are fatal. when there's a mutation in the viral genome, it can cause the loss of replication capability. However, retroviruses are produced from infected cells very, very rapidly. If you produce 1,000 viral particles, if most of the mutation is fatal, still 1 in 1,000 can be beneficial for the proliferation of retroviruses. In addition to that, retroviruses can acquire host gene into the viral genome. How? As I told you, retroviruses reverse transcribe the viral genome into DNA. So during the process of reverse transcription, if there's an host cell RNA that has some homology between viral genomic DNA, the reverse transcriptase can produce a DNA strand, minus sense DNA strand from the viral sequence, and at the point of homology can switch the lane from viral RNA to cellular messenger RNA, and then go back to viral RNA sequence through the process of homologous recombination. And by that way, during the replication, retroviruses can acquire cellular gene into viral genome. So the one consequence of that is, for example, BIF protein that is encoded by HIV. HIV BIF is a viral infectivity factor. This factor, viral infectivity factor, BIF, is not required for the replication of HIV as a retrovirus in cultured cells. As long as you are handling HIV in cultured cells, the function of BIF was unknown. Why HIV contains this BIF gene while it is not required for the proliferation of virus in tissue culture condition? But if you infect the primate cells, body, monkey, or the macaque, both individuals with SIV, HIV retrovirus, without BIF, those viruses do not replicate well in vivo. Why is BIF required only in vivo? Because BIF protein binds specifically with host cell ApoBIF3 and induces the ubiquitination of the host cell ApoBIF3, leading to the destruction of the host cell ApoBIF3, so that the BIF-containing virus can expel ApoBIF3 from budding virions. And this process must be, of course, species, very, very species specific, because the ApoBIF3 sequence, amino acid sequence, can differ from one species to another. So what is known is human ApoBIF3 is destroyed by human retroviral HIV BIF, and African green monkey ApoBIF3 is destroyed by SIV African green monkey AIDS virus BIF, and macaque ApoBIF3 is destroyed by macaque AIDS virus BIF. So there's a very, very strong species-specific interaction between BIF and the host cell ApoBIF3G. So what is believed until about 2008 was that we acquired ApoBIF3, but the virus is more rapid in evolving and acquired BIF that inactivate our ApoBIF3. And by the duplication, gene duplication, we acquired new ApoBIF3 family protein. But at that time, the retrovirus acquired the new BIF protein that neutralized the restricting activity of the newly acquired ApoBex3. And so we acquired new copy of ApoBex3, but the virus emerges with the mutated BIF. So that many researchers were believing that we were losing this fight against the retroviruses, and currently disease-causing retroviruses, HIV, is resistant against our restriction mechanisms. But we found this to be incorrect in the case of mice. Okay, so by using friend mouse retrovirus, we were analyzing the host factors that works to restrict the replication of retroviruses and restrict their disease-causing processes. So what is friend retroviruses? Friend retrovirus. It sounds like very friendly virus, but it's found by an American researcher named Charlotte Friend, and it is actually a very, very nasty virus. And when infected with this friend virus, it causes the enormous expansion of the spleen cells. And in the spleen, the red blood cell producing progenitor cells, erythroid progenitor cells are expanding very rapidly, causing the 20-50 times enlargement of the spleen. And infected mice can die within 2 months by showing the enormous infiltration of erythroid leukemia cells, not only in the spleen, but into the liver and the brain. So how this virus causes the disease? Of course, friend virus is a retrovirus, but friend virus is a complex of two different viruses. One is usual retrovirus that contains the RNA genome and all the structural proteins, as I told you, the core producing GAG and reverse transcriptase encoding PO and the envelope gene. On the other hand, the other component of the friend virus is a so-called spleen focus forming virus, and it is defective. It does not contain all the structural genes, but it contains a defective envelope gene, the product of which is expressed on the surface of the infected cells and interacts with erythropoietin receptor and causes the proliferation and differentiation of infected cells into erythrocyte, red blood cells. And this protein GP55 encoded by the spleen focus forming virus M gene is also interacting with the tyrosine kinase type receptor expressed on the hematopoietic cells, and it causes the rapid growth of hematopoietic cells. So what happens is when infected with friend virus, the erythroid cells are proliferating very rapidly due to the stimulation through the erythropoietin receptor and this hematopoietic cell specific tyrosine kinase. And proliferating cells are reinfected by friend virus, either SFFV component or friend helper virus, the replication competent component. And the repeated integration finally causes the expression of PU1 gene that is a key molecule in regulating the differentiation of hematopoietic cells. So growing, proliferating hematopoietic cells stop differentiating due to the induced high expression of PU1. And finally causes leukemia like this. But not all strains of mice are susceptible to friend virus infection. Some mice are resistant. For example, B6 mice, black 6 mice, are very, very resistant to friend virus infection because it has the B6 mice have a mutation in this tyrosine kinase gene product, and the growth induction signal is not well transduced into the infected cells. So B6 mice are believed to be very, very resistant to friend virus infection. Now, as a matter of fact, B6 mice do not develop any fatal disease after infection with the friend virus complex, while as I have shown, the susceptible strains of mice are dying very rapidly after infection. However, if CD4 T cells are depleted from otherwise resistant B6 mice, they start to develop the disease and showing the enormous big size of the spleen. When B cells are depleted, however, there was no disease development. And it means that the B6 mice are resistant to friend virus infection due to the immune responses mediated by CD4 T cells. So we have identified several different genes that control immune responses to friend virus infection in the B6 mice, and we mapped them into the different chromosomal segments, and many of them are, of course, located within MHC. In the case of mice, it's H2, and those genes are controlling, as I show here, T helper cell responses or NK cell responses or cytotoxic T cell responses. However, there's a gene named RFV3 that is not located in the MHC area, meaning that it is not controlling T cell responses, but interestingly, it controls the responses, antibody responses to friend virus infection. And that gene is mapped in chromosome 15. And all other genes are identified, molecularly identified, and their mechanisms of action have been clearly shown. But this gene, RFV3, had been unidentified of its mode of action. So what we found was, this is what we did a long time ago. In resistant strains of mice like B6, when they are infected with friend virus, they rapidly produce neutralizing antibodies. This is the titer of neutralizing antibodies, okay? However, the susceptible strain like AJ or AWISN do not rapidly produce neutralizing antibodies. They cannot produce neutralizing antibodies until 3 weeks after infection. So we crossed this antibody-producing strain with a non-producing strain. A is non-producing strain, and B10 or B6 is producing strain, and found that the F1 progeny can produce antibody rapidly, meaning that there's one autosomal gene that is controlling the production of the neutralizing antibodies. So we back-cross. This is a very, very classic Mendelian genetics, okay? You know that? This is the recessive strain A that cannot produce neutralizing antibodies. Since the production of neutralizing antibody is a dominant trait, F1 mice, the producer strain and non-producer strain F1 can produce neutralizing antibodies. And finally, back-cross them to non-producer strain. Half of them produce neutralizing antibodies at 2 weeks after infection, but half of them did not produce neutralizing antibody. So we did all genetic analysis and found that when the back-cross mice possessed B10 or B6 derived the genetic marker in chromosome 15, they produce neutralizing antibodies. If they lack the B10 or B6 derived genetic trait at this location in chromosome 15, they do not produce. Okay? So this is a classic genetic mapping experiment, and we finally narrowed down the area of the chromosome which harbor the neutralizing antibody controlling gene into this short segment of chromosome 15. And in the center of that candidate area, we found ApoBec3. And we actually found that mouse ApoBec3 gene is very, very polymorphic and differs its expression levels and amino acid sequences between susceptible and resistant strains. So here, resistant strain express higher level of ApoBec3 comparing to the susceptible strains. And there's an amino acid substitutions between the resistant ApoBec3 and susceptible ApoBec3. And interestingly, the resistant ApoBec3 lacks this area. This is encoded by exon 5. Okay? So, there are several differences between the product of ApoBec3 encoded by resistant allele and susceptible allele. One is higher expression level induced by the presence of the resistant ApoBec3 allele, and there are amino acid substitutions, and there's a differences between the presence or absence of exon 5. So which of these are actually associated with the observed susceptibility or resistance to friend retrovirus infection? So what we did was to produce knockout mice, of course. And when ApoBec3 is knocked out from otherwise resistant B6 strain, the knockout mice showed more than 100 times higher viral replication. This is the log scale, okay? So more than 100 times higher viral replication, both in the spleen and bone marrow. And this activity of the B6-derived ApoBec3 allele cannot be substituted by the susceptible allele. Here, the knockout mice are crossed with the susceptible strain BARC, and when the resistant allele is lacking, even in the presence of susceptible allele, we observed 100 times higher replication level of the retrovirus, friend retrovirus, and it is associated with the development of much severer diseases. As you can see here, the number of infected cells much increased, and because of the induction of erythroid cell proliferation, we observed a higher hematocrit level in the peripheral blood. And the higher expression level, as you can see here, of the resistant strain is very interestingly associated with the presence of endogenous retrovirus. In resistant B6 strain, there's an insertion of endogenous retroviruses, endogenous retrovirus within the intron between the second and third exon. This is very interesting, isn't it? The target of the restriction itself, when it is inserted into the resistance-associated gene, it causes higher expression of the resistant-associated molecule and induces the restriction against the retroviral infection. So ironically, the ancient retroviral infection into B6 strain caused the higher expression of restriction molecule. So we are utilizing our enemies as our allies to fight against retroviruses. This is a very interesting example. Another thing that is associated with the difference between the B6 type and BARC type allele is the presence or absence of exon 5. So this is a little bit complicated slide, but what I'm showing here is the product of resistant allele that lacks exon 5 is more stable as a protein than the exon 5 containing susceptible, the product of exon 5 containing susceptible allele. So as you can see here, the exon 5 lacking the resistant associated protein is produced much earlier than the exon 5 containing product, and the level of expression is much higher comparing to the level of expression of resistant protein is much higher than that of the susceptibility associated protein. And this is caused by more efficient translation of exon 5 lacking messenger RNA because when the messenger RNA contains exon 5, it causes more stable higher order structure of the messenger RNA. It impedes the translation of messenger RNA into the protein. And we wanted to understand why the product of the resistant allele is expressed higher. I mean, why the resistant allele does not include the exon 5 into its messenger RNA. To do that, this is a very professional experiment, but we did an in vitro splicing, in vitro translation of cloned DNA into messenger RNA, and we made chimeras between resistant associated and susceptibility associated allele and expressed them in vitro and let them transcribe messenger RNA. And to make the long story short, our conclusion was that the single nucleotide substitution observed within exon 5 is responsible for the presence or absence of exon 5. For example, here, if we have the substitution of G to C at position 88 of the resistance allele, it induces the inclusion of exon 5. So let's go further. So we wanted to know how the resistant strain B6 or B10 acquired this resistant associated genetic trait. Okay, so we have susceptible strains of mice, laboratory animals, and resistant strains of animals. And we know that, now we know that only one single nucleotide substitution is responsible for the inclusion or exclusion of exon 5. When exon 5 is included, the protein translation is inefficient. When the gene does not include exon 5 into the messenger RNA, higher level of protein is produced. And what we were expecting was that, okay, ancient mice are all susceptible to retrovirus infection, and the ancestors of currently resistant strain like B6 by chance acquired this single nucleotide substitution and started to produce large amount of restriction protein. So to test that, we sequenced all different wild mice. Okay, so there are many different species of mice that can be analyzed for genetic analysis by catching them in the field, and we compared the ApoVec3 sequence of many different species of mouse genera, and what we found was unexpectedly ancient ancestors this is a phylogenetic tree, and what we found was ancient ancestors of current mice were all possessing this high expression associated genotype. Okay, they all had this G here instead of susceptibility associated C. So ancestors of mice were expressing larger amount of resistant factor, and somehow some ancestors of current susceptible strain acquired genetic change that caused lower amount of resistant factor expression. And as a matter of fact, this exon 5 that is associated with lower amount of protein expression is actually dispensable. And many wild species of mice, including rats, have a frame shift mutation, deletion of one nucleotide or addition of one nucleotide that causes the frame shift, the shift of the reading codon, and that causes eventually causes the presence of stop codon within the coding sequence. So for ancestral mice, ApoBec3 was very, very important, and they expressed large amount of the resistant factor protein, but somehow some ancestors of current susceptible strain acquired genetic change to include exon 5 that causes lower amount protein expression. This is very, very unusual. How did it happen? It's known that the ancestors of mice, current mice, are born in Indian subcontinent. And those ancestral species of mice migrated into Eurasian continent in taking two different paths, two different routes. One route is through the eastern coast of Caspian Sea and going up to the Siberian area, and they distributed in northern Europe. The other route is going through the Arabian area and go through the Mediterranean, and they distributed to southern Europe. Okay, so what we found was ancestral species of mice expressed large amount of ApoBec3 protein, as you can see here, and they kept that ability through the migration to the north, going through the east coast of the Caspian Sea. And during that process, they acquired endogenous retrovirus. Okay, and the acquisition of the endogenous retroviral LTR within the ApoBec3 gene induced much higher transcription of the already efficiently transcribed messenger RNA. So this acquisition of LTR, endogenous retrovirus, caused the expression of restriction factor protein much higher. On the other hand, some of the ancestral species somehow acquired genetic change that caused less efficient translation of messenger RNA into the protein and very small amount of protein expression. So why this kind of seemingly meaningless, seemingly deleterious genetic change was acquired during this migration? Okay, so here's the secret. As I told you, we were attacked by retroviruses coming from different species, and it's known that xenotropic viruses came from the primates and cats into mice. And when we tested the presence of endogenous XMV is xenotropic virus, okay? Endogenous xenotropic virus, what we found was those migrated through the east coast of Caspian Sea contains large copy numbers of xenotropic virus. So ancient xenotropic virus infection caused the preservation of the highly expressed ApoBec3. They needed this highly expressing trait to fight against repeated attack by xenotropic virus. And one of the integrated xenotropic virus caused even higher expression of this resistance factor. On the other hand, as you can see here, okay, mice migrated through the Mediterranean coast do not have endogenous xenotropic virus. So they were not attacked by xenotropic viruses. Therefore, they didn't have to keep the high expression of this restriction factor. But what was the advantage of reducing the expression of ApoBec3? It's known that when ApoBec3 expressed in a higher amount, it can cause the relocation of the presence of ApoBec3 into nuclei. So it can cause the mutation of the cellular DNA itself. And it is already known that some human cancers, like breast cancer or uterine cervical cancers, are associated with the clusters of mutations that are seemingly caused by ApoBec3 function. So one hypothesis is the mice that migrated through Mediterranean coast were not attacked by xenotropic viruses, and because of that, they wanted to preserve their genome by reducing the expression of otherwise possibly deleterious DNA mutator ApoBec3. But these strains, ancestors of currently resistant strains, had to keep the high expression of ApoBec3 because they were continuously attacked by xenotropic viruses. How? Yes. Time? Okay. Okay. So I go to the conclusion. Okay. So mouse ApoBec3 is physiological functioning. This is what we showed first. And it's capable of restricting retroviruses. And mouse ApoBec3 is polymorphic, and resistant strains express higher levels of messenger RNA. And the higher level of expression is caused by endogenization of the enemy. Okay? And mouse ApoBec3 exon 5, if it is included, it interferes with the transcription and results in the lower protein expression. And I didn't have time to talk about humans, but the similar thing is known among humans. And the humans stopped expressing high levels of one of the copies of ApoBec3. And in mice, presumable selective pressure for functional exon 5 lacking ApoBec3 would be xenotropic virus that are coming from other species into mice. So thank you very much, and I thank grant support from Japanese government and my university, and this is my laboratory members, and this is when Virginia visited our laboratory. Okay, thank you very much.